Friends, today I'm going to share the notes on the RNA editing process. So, editing is a mechanism by which the RNA transcribed RNA are changed itself. And it is widely accepted that the base sequence of mRNA represents only the sequence present in the DNA from which it has been transcribed. So the RNA editing may be confined to a single gene, or it may be the effect of entire mRNA. And the mammalian RNA liver cells, which contains apolipoprotein B, having 4,563 amino acids, while in the case of intestine cells, this protein has only 2152 amino acids. A relatively small sequence of 26 bases surrounding the editing site provides a sufficient target for editing to occur. The RNA editing enzymes are related to the general nucleoside deaminases, but these enzymes have other regions or subunits that determine their specificity. More dramatic changes in the mRNA base sequence have been found in the trypanosoma, mitochondria, and the SB5 and measles paramyxovirus. But in trypanosoma bacteria, the COX2 mRNA has an addition of 4 U residues, that is uracil residues. That this is not only the introducer additional amino acid but also produces a frameshift beyond the omit of insertion. A similar dispermanency, that is lapse between the DNA and the corresponding mRNA sequence, is found in the SB5 and the measles paramyxovirus, but in this case the G residues, the guanine residues, are added in the RNA. In the case of gene of COX3 of the T. bruceli is extraordinary case. Here more than half of the residue in the mRNA consists of uridine that are not encoded by the genes, and in addition the flanking region on either side of the sequence are complementary to the sequence and they re-edit. Guide RNA are encoded by separate genes from those that encode the mRNA which then edit. In the case of Leishmania mitochondrial genome, the genes coding in the pre-edited RNA are interspersed with units that encode the guide RNA required to generate the correct mRNA sequence, because the certain genes have multiple guide RNA. The pairing is more extensive on the 3' end of the site of the target site than it is on its 5' site. The unpaired A residues, adenine residues, specify the sites of the addition of the uracil residues in the target sites, and the endonuclease cleaves the substrate RNA at a site that is presumably specified by the lack of pairing between the guide and the substrate RNA. The TUT which is an enzyme, adds the uracil residues from the UDP to the substrate RNA at the site of the cleavage, which is later on ligated by the RNA ligase activity. So it is a short notes on the RNA editing process. Thank you and keep watching.